Kreft from the University of Birmingham here in the UK, and he's done some beautiful work on individual-based models of biofilm. But I think today he's going to talk about models more generally, whether simple models are more general. I don't know if you need to do something with the microphone.
Is the mic working? Can you hear me? Okay, if that's better. So basically what I'm going to do is today is, because it's a summer school about methods, I thought I should say a few things about modeling, individual-based modeling, give a bit of advice or raise some common problems. So basically this is going to be a rant of an angry man, and I hope you excuse that. So I will do a little bit of an introduction into individual-based modeling, but first want to thank all the people involved in the development of the software dynamics that we've been using for a couple of years now. Originally written by Laurent Ladong and Brian Murky at the DTU with Bart Smets postdocs, and now more developed by Kieran Alden and Robert Clegg more recently. And iDynamics we also are now using to turn this into a model of the electronic gut. So this is my new project on developing individual-based models funded by the NC3Rs as a replacement for animal research. So this is a 3-year project to turn an individual-based model of biofilms and chemostats into one larger scale model of the gut. So individual-based modeling is about interactions between the microorganisms. So basically you have cells here, different sizes, different positions, different colors. They may interact or may not interact with each other. If they are, this may depend on the distance, and if they are getting too close, they will certainly mechanically interact, but that's nothing, no specific interactions. They will also interact with the environment. The environment might be spatially structured. And maybe all the interactions between the cells are indirect via the environment. So out of all these interactions, you get the population or community dynamics and population structure, biofilm structure, as emergent properties. So emergence is a very important concept that I hope I'll explain a little bit more in the next few slides. You can make, in an individual-based model, you can make the cells more very simple or quite complex. That's up to you. It depends on the purpose of the modeling, what's a sensible choice. So you can make the cells complex systems biology models themselves with lots of intracellular dynamics, or you can make them very simple based on growth kinetics and rules. That really depends on the data you have and the question you have. So just to reiterate the point that in an individual-based model, you really only describe the activity and behavior of the individuals, and you describe what happens in the environment, nothing else, because everything else is in emergent properties. So you have the feedbacks between the organisms, or you have competition for resources as an emergent property, and not something you have to specify in the model. So in an individual-based model, you do not specify competition. You specify two organisms eating the same resource, and the competition is automatic. That's a very important point to understand. Because of that, fitness itself is also, of course, an emergent property. Another thing that is important to me to point out is that individual-based models are structurally realistic, and that's what attracted me to individual-based models in the first place, because they represent the model represents reality in a way that's easy for us biologists to understand, because we're talking about individual organisms in the model, and we know that they exist, and we know the environment exists, and so on. So it's this close similarity between the things in the model and the things in reality. So when I was saying that individual-based models are about individuals, that's only half the truth. The other part is, of course, the environment, and the environment actually is usually described by partial differential equations. So it's really a hybrid between agent-based model for bacterial individuals or microbial individuals and partial differential equation model for the environment. And these are, of course, coupled. You may have seen this famous photograph of a Petri dish from Alexander Fleming. And if you're thinking here of this organism growing into a colony, producing some compound that diffuses and inhibits the growth of these colonies here. You can, in principle, study the growth and production of the toxin by this organism in a pure culture without these. And you can study the growth of these and the effect of the toxin on these organisms in another pure culture, and then put this information into a model, and the model will then predict the interaction between these two based on your study of them in isolation. So the point here is that this interaction again is emergent, and you can study this in isolation, and the model can synthesize this information and recreate the interaction. So I think this is very important if you're thinking about the problem that, if it's a problem, of the extremely high biodiversity in the natural environment. If you cannot possibly study all the interactions between all the organisms in the environment, there's far too many, and it leads to combinatorial explosion, where very quickly you cannot study in the lab all the possible combinations of interactions. So having a model that allows you to predict interactions based on studies in pure culture would be very useful for this. Okay, so a few things that you can do with individual-based models. First of all, here in this case, this is an image of a flow cell biofilm, and you can use this image to generate an individual-based model that has the same biofilm structure, and then using known kinetics of the substrate consumption of this organism here, you can then recreate in the model the spatial concentration gradients. Okay, this is based on an experimental biofilm structure, and you can then generate the concentration gradients in the model. You can validate this with microelectrodes, and here in this case, you're not reading in a biofilm structure, but you're predicting a biofilm structure and the gradients in the biofilm structure based on studies in a chemostat. Thanks for introducing the chemostat previously. So you can use growth kinetics that you can measure in the chemostat in order to endow all your individuals with growth kinetics, and they will of course grow at different rates given different substrate concentrations. And using diffusion reaction equation, you can predict the substrate concentration gradient and therefore also the growth rate gradient in this biofilm, and the biofilm structure itself is also something that the model generates, not something you have specified in the model. So you can basically use a model to predict what would happen in a biofilm, for example, based on data in a chemostat, and the extent to which if you really understand the dependence of the growth of the organism on substrate concentration, you should be able to predict what happens in the biofilm. If that prediction fails, then you know that you are missing some information that is important. You can also construct synthetically communities, for example here interspecies hydrogen transfer, where you have a hydrogen producer and a hydrogen consumer producing or consuming hydrogen, and you can study this in isolation again and put it together in a model, and it will predict the rate of hydrogen transfer in given spatial positions of the cells. So the model basically is able to translate data generated from simple systems into more complex systems, and I guess that's a useful thing to be able to do. Now, individual-based models have one advantage, that they can make use, of course, of data on the individual level, but they can also use data on the population level to compare with model predictions, and because of that they can make use of more different types of data than population level models, because the individual-based model maps the individual level to the population level, so you can use more different types of data to inform the modeling. Okay, a few things that you should not do doing individual-based modeling or similar modeling. You should not impose the behavior of the individuals that you are interested in studying. If you're forcing your individuals to do something, if you're trying to force or nudge your organisms in the model to produce certain structures you're interested in, that's not a good idea. If you really want to learn how these structures form, you shouldn't prescribe it in the model. And there's a certain tendency that people do that. Another tendency is that they give their agents global information. For example, I've recently reviewed a manuscript where they were specifying the organism was moving in a biofilm situation to the highest position in the biofilm. Now, of course, a microorganism cannot measure height in a biofilm. It cannot know that. It can sense concentration gradients or things like this, but an organism cannot sense height. Right? So making your organism in the model do things like this is bad. And it's also trying to prescribe that organisms move upwards. This is again something you shouldn't do, because then you can't be very surprised that actually the organisms in the model move upwards, right? If that's what you're forcing them to do. So where is the gain in that? But this is sort of commonly done, and I've reviewed many papers with things like that. So what you really should be doing when you're modeling microorganisms, you should put yourself inside a microorganism and think about what life is like in your system from the point of view of the microorganism. As a modeler, you know everything about the system, but the microorganism in your model doesn't have that information. So you really have to think from the point of view of the microorganism, not from the kind of observer point of view that you have. Okay, one thing that often comes up in when you get individual-based models reviewed, that the reviewers complain about them as being too complex or so, and sometimes that might be a correct complaint. It's actually not so simple to say that individual-based models are more complex. In some sense, they are actually quite simple. They are conceptually simple, by which I mean that you really the idea of just describing what individual organisms do, the activities that they have, and to describe what happens in the environment is conceptually very simple. Sometimes also the activities of the organisms can be very simple in these models. So the model can have very simple input and can be more complex if you're describing a lot of activities, or there's a lot of intracellular dynamics that the model has. Then the input can also be complex. What they certainly are is that they are computationally complex. So the software, the code can be quite complex because it has to keep track of all the individuals and make sure that they're all stepped in random order and so on and so forth. So this is an area where individual-based models are difficult because the software is complex and difficult to test and difficult to describe. Thankfully, there is a standard called the ODD protocol for overview design principles and details that helps people to think carefully about how to describe a model and what you need to say about your model that other people can understand what really goes on in your model. So this was invented by Grimm and co-workers and has proved very useful for communication of these computationally complex models. The output of the model may also be quite complex in terms of complex structures of biofilms, for example, complex patterns. but this usually is a result of simple input. So IBMs are both simple and complex in some sense. Because the whole workshop here at the Newton Institute is about understanding microbial communities, and the complexity of these communities is maybe the major challenge for us. So I thought I should have one slide about how you can deal with the complexity, and of course the answer is kind of obvious. You have to simplify, right? If it's very, very complex, you have to simplify somehow, and that is obvious. But how to simplify is not that very easy, because this I think is a very nice quote. Simplifying assumptions are both the soul and Achilles heel of mathematical models. Very true. So this is not an easy area. How do you simplify? You can simplify biodiversity, maybe by substituting all the strains and species with functional groups. And that certainly allows you to cut down enormously on the diversity. It may be appropriate, but maybe you're interested specifically in one or two organisms, and then you might want to include them in detail and have the rest of the community, sort of the background community, at a coarser level, just in terms of functional groups. So it's one thing you can do to simplify biodiversity. You can also simplify spatial structure by making assumptions about mixing in the system being fast relative to the time scale of whatever you're interested in. And you can simplify again by making the steady state assumption. One of the greatest inventions, I think, in mathematical modeling is the steady state. It really helps enormously to simplify systems. The alternative is to measure a lot and have a lot of information, a lot of data that will allow you to have more complex models and still be sensible to do that. Okay, so that was the introduction on individual-based modeling. Now I want to give you an example of some recent work in my group as an example really of the problem that if a model is too simple, it may actually be less general. There's usually the idea that simple models are more general, and I think this is an inheritance from physics, where we have very general models that are also very simple, but biology isn't physics. So this is about aging in unicellular organisms, work done by my excellent former PhD student, now postdoc Robert Clegg, was helped by Rosemary Dyson from the Maths department. And the question we looked at was the question why aging evolved, although obviously it's a disadvantage if you die. Okay? So first of all, going back a long time to the germline soma distinction in multicellular organisms, not all organisms, but many multicellular organisms have this distinction. This is very important because this allows a division of labor. And the reasoning from Peter Medawar that early reproduction is favored by evolution because there is such a thing as extrinsic mortality, because there is always a chance that one dies. Even though one might be able to live forever, at one day we're going to die, right? So because of that, extrinsic mortality, there is an advantage in reproducing early, because then you're more likely to have reproduced before you die. So, and these ideas have culminated in the current theory explaining the evolution of aging, the disposable soma theory by Tom Kirkwood. Now with unicellular organisms, which is what we were looking at, this distinction doesn't really exist between soma and germline, or maybe. maybe there's some more subtle division of labor possible even in unicellular organisms if they divide asymmetrically. That's very important. If the cell division is functionally asymmetric in some sense, that there's more damage going into one of the daughter cells than the other, then you have some kind of division of labor even in unicellular organisms. So you need a mechanism, segregation of damage, and you need an evolutionary advantage for this to evolve. Just a few experimental evidence. Caulobacter crescentus is a very interesting bacterium that has that is attached to a surface by a stalk, and it produces swarmer cells that are motile and swim away and colonize the surface somewhere else. One to grow, one to go. And this organism has this strange way of cell division, which is budding. So the swarmer cell is budding off, first very small bud in growing bigger and then leaving. And this organism, if you look at the reproductive output of this stalked cell, it goes down with age quite strongly. So there is clearly asymmetric division in this organism. And a bit later, 2005, Stewart et al. Showed in E. coli that there's also a little bit of asymmetry in division, where the younger daughter cell has a slightly increased growth rate from generation to generation, and then the slightly older mother cell. So there is a possibility of asymmetry in cell division of unicellular organisms. But there is a big difference between the budding Caulobacter and the fissioning E. coli. So budding, I think, is fit for the purpose of segregation of damage, whereas fission is less fit for purpose, where the segregation of damage is kind of accidental. If you have a few protein aggregates, they are more likely to end up in the old pole cell than the new pole cell because of passive diffusion and restriction of diffusion by the nucleus, so no active mechanism. Our question was whether this is beneficial to have this damage segregation, and that of course is a question of whether there isn't a better alternative. So there have been some mathematical models, quite a number, asking this question. One model by Martin Ackermann et al. Is conceptually very simple. You have a cell with a certain amount of damage, which gets a new amount of damage every generation k. So you end up with a certain amount of damage, then some is repaired with rate r, and then divided symmetrically or asymmetrically into the daughter cells. And now the survival of the daughter cells, as survival depends on the damage and the repair rate. So survival goes down with damage, makes sense, and survival goes down with repair rate. This is a way to implement the cost of repair in the model. Now that's of course very important that you have a cost for repair, and this is the way they have done this. So there's a cost of repair, and the advantage of repair, you have less damage, so the survival is increased. And the simulation results are that if you don't allow asymmetry, repair evolves, and if you then allow asymmetry to evolve, repair evolves to 0, and asymmetry evolves to 100%. So a very clear answer. Asymmetry is perfect asymmetry is optimal, and 0 repair. Now Erzsébet a little bit later published another model with similar conclusions, but they actually ignored repair completely. There's no repair in their model. They also have no external mortality, as Ackermann had. Resource competition, none of them had. Erzsébet actually also looked at different sizes of the daughter cells, showing in the model that if the daughters are smaller than the mother, then asymmetry is not beneficial. which is in total contrast to the experimental results of the budding yeast, which has asymmetry and has has damage segregation and has smaller daughter cells. So the model is actually predicting the opposite of what's observed in the budding yeast. That didn't seem to bother them. This is one set of equation which they've called the linear growth rate, because there is the increase of intact protein doesn't grow with isn't proportional to intact protein. So they have called this linear growth. Actually, intact protein inhibits growth, which is really I don't understand how you can possibly set it up in this way. The second part of their model is what they call exponential growth, because there's p int here in the numerator, but actually this, if you look at the parameters, ks is 1 and p int p dam is around about 750 to 1500, so ks is practically negligible. So you end up with the ratio of intact protein over total protein, which is youth, or 1 minus age. So what that means is actually that the rate of increase of intact protein is linear decreasing with age and constant. So it's not exponential growth, but in their model description they call this exponential growth. I don't really know how this gets through review. And the predictions were also not correct. Anyway, so in our model, what we do is we are starting with a normal model where cells grow by taking up substrate and converting it into active protein or biomass. And if there is enough biomass, the cell expands and divides. And because all cells take up substrate, you have automatically resource competition in this model. So it's a basic model of growth, and on top of that, we have added the process of damage. So the active protein can become damaged, and the damaged protein can become repaired. But in order to repair the damage, you have to have some repair machinery. So the active protein is split into the part that catalyzes growth and the part that catalyzes repair. And the damaged protein may or may not be toxic. So repair in our model also is costly because it takes away protein, active protein from the growth process. And that's very important that you have a cost for this in the model. And the cell division is, as usual, either symmetric or asymmetric or somewhere in between. I don't have much time, so I only show one slide of result, which really summarizes this all. If you first look at inert damage, you can see that the symmetric cell division, so no damage segregation, is always better than the asymmetric division. And you see for toxic damage that the effects are much stronger, of course, and that you have, again, an optimal investment into repair machinery. And if at this optimal investment into repair machinery, for example here, the symmetric cell division is better than the asymmetric cell division, which also has an optimum which may be somewhere else. So repair is better than segregation of damage. And this is true for a very wide range of parameters. Only if the damage is toxic, accumulates at very high rate, and the efficiency of repair is low, do you get an advantage in damage segregation, but on top of repair, not instead of. Okay, so why does our model make the opposite predictions to previous models? I think the key reason is, so we have also re-implemented Erzsébet to look at this question, but the key reason is that in the model by Ackermann, the cells are not actually growing. They are stepped every generation and divide every generation, but they don't grow. In our model, if repair returns damaged protein into active protein, this allows the cell to grow faster immediately, resulting in a cell division earlier if the cell grows faster. But in this model, the cells all divide at the same time. So there's no immediate advantage in the repair as in our model. So our model has an immediate disadvantage of repair by taking away active protein, but also has an immediate advantage in repair, and that is a difference. It's a small difference, but it has strong effects. So, but we should compare our model to evidence, of course. So the evidence is our model predicts repair to be universal because it's always beneficial. Okay? So all organisms should have repair processes. And as far as I know, all organisms that have been studied do have mechanisms of repair of damage and mechanisms of preventing damage. So that prediction is correct. Our model also predicts that damage segregation is not beneficial under benign conditions in lasting environments. Now there is evidence, for example, basically the budding yeast and the budding bacterium Caulobacter are the two organisms that show strong aging. Okay, and that appears to be in contrast to our model prediction. Then there are many other microorganisms like E. coli and others that show actually minimal aging if they are grown under benign conditions. Also the fission yeast shows minimal aging under in the absence of stress. So what's important to notice is that there is a big difference between different microorganisms in how strongly they age. If the model would predict that 100% damage segregation is always best, all organisms should strongly age. But the fact is some organisms age very little and some age more. So in order to explain these differences in the observations, we think that you need to consider the ecology of the organism, because if the organism lives in a short-lived environment, there's no point in being long-lived yourself if the environment is not long-lived also. And that is true for these two organisms here. They live in short-lived environments. Budding yeast spoils the environment by producing ethanol and makes the environment short-lived. And Caulobacter attaches to surfaces which are only transient and be eaten within a few days by nasty ciliates or other organisms. And these organisms here also have a budding mechanism. So it's a combination of the budding mechanism that allows a stronger asymmetry in division and combined with the ecology of the organism living in short-lived environments, which I think allows us to explain all the experimental evidence and make this consistent with our model prediction of no aging under benign conditions. So if aging would actually be beneficial, as previous models have suggested, all organisms should really be budding. But that's not the case. Some bacteria do bud, but not that many. Okay, so this is just sort of the little bit of science I wanted to present today to give you an example of different models producing totally different predictions, and the models without growth, without repair, making very different predictions. And while I'm at it, I think this is another big problem in science, that if the fashion is that everyone is looking for aging in all the organisms, because that's currently what people think is interesting, people go to some lengths to prove aging in their organisms. So here's a paper on diatoms showing that diatoms also age, that means favor their younger daughters. And these are the results. The fact that this slope is significant is important for the conclusion of the paper. But I didn't quite believe that, so I scanned in the data and did the regression myself, and I found that this one data point here has an extremely high influence on the regression parameters. if you can measure this with Cook's distance. This is 50% of the entire Cook's distance is in this data point. That's 1 out of 82. If you take that out, the regression loses significance. So the conclusions of this paper hinge on a single data point out of 82. So, yeah, if you're trying very hard to find what you want to find, you will. Okay, so I'm trying to come up with some general conclusions beyond this aging work. The problem with some of the previous models were that they lacked basic processes like growth and or repair as an alternative solution to aging. So they were too simple in a sense to be general enough. So that's what I mean by too simple, lacking basic processes of growth, because this including growth process in the model is not a problem, because we know this happens. It's not we're not making assumptions about things that we are not certain about. So, and I think this is a really pervasive issue, because obviously every one of us doesn't have infinite time to develop a model. You may have a 3-year project, and you cannot do that much in 3 years. So if it takes you half a year or a year to implement a process just to test out whether it's important or not, you can't do that. So you end up with a model that hasn't tested all the processes that might potentially be relevant just because you can't do it. No one can. So I don't blame anyone about that. But I think there's one solution to this, and this is basically when we were to develop the code for a modeling platform as a community development where everyone is working on a particular process like aging or signaling or plasmid transfer and contributes the code, then the model becomes powerful enough that you can simply, within a few days, test out all kinds of processes, whether they are relevant for your particular question. And then you can be sure that you're not missing anything. Okay, so I think we really need to get away from everyone developing their own model for one specific question and therefore ignoring lots of other things, and someone else ignores other things, and no one has a model that has the capacity to try out all sorts of processes like signaling quickly. You heard Sam earlier talking about quorum sensing, for example. So that's another. This is probably a quote you've come across. Has anyone knows that quote? There are 220,000 hits on Google with that quote, so you may have come across this by George Box. So what I don't like about this is that he says all models are wrong without making any sort of distinctions of how wrong, because it's not just wrong or right. That's too simple. Okay, so obviously models can be wrong. You know, the model that the Earth is a flat disk is wrong, totally wrong. Okay, so models can be totally wrong, and models can be not quite so wrong. And this is the kind of models we're interested in, that are not quite so wrong, that you wouldn't want to call them totally wrong. Okay, so the predictions, of course, should be correct if the assumptions are correct, because a model is just a logical machine turning assumptions into predictions. So if all the assumptions are correct, the prediction necessarily has to be correct. That is the idea is correct in principle, but the problem is you never know exactly. You will always know exactly the assumptions you're making, but not the assumptions you're not making, or your way you're basically assuming this and this and this and this is not relevant. Because these are assumptions, which is basically a long list of things you're not considering. And in a sense, these are assumptions, but you're never clear about the whole list of assumptions you're making. So there can be problems with, so you can end up with wrong predictions although all the assumptions you have looked at are correct, because you have not a complete list of assumptions, you never will be able to have a complete list. Okay, so what I'm trying to say is that models can be less wrong than others, and that's actually what we should be trying for. And I would want to rewrite the quote from Box to say that models are more useful if they are less wrong. That actually a model cannot be useful because it's wrong, but if it's useful, it's because it's not totally wrong. Okay, some more examples of models where there are some problems. This is a model of mutualism in the gut. You have two species here, and they compete, and the host, this here at the bottom is the host epithelium, can favor one or the other species by inhibiting the other, or by feeding specifically the one it wants to favor. And what this paper has shown, that is fairly easy for the host epithelium to favor one strain over the other by selective feeding or selective inhibition. And all the cells here in this model, they start to grow at the bottom near the epithelium layer, and they grow upwards, and there are no bacteria here at the top where the lumen of the gut is. And they show a picture of a gut where actually there are loads of bacteria in the lumen and hardly any in the epithelium. So this is in stark contrast to this situation here, where all the bacteria are close to the host mucosa and there are no bacteria in the lumen. So by assuming in this model that there are 0 bacteria in the lumen, and therefore there is no exchange of bacteria from the lumen into the mucosa, you're basically assuming that the dynamics in the lumen and competition in the lumen play no role. And that may be true, I don't know. I haven't done that. So this may be right. But it's in stark contrast to the fact that actually in the real gut, the lumen is full of bacteria. And the mucosa is actually depleted in bacteria, especially close to the host epithelium. You do not find many bacteria, as this image in that paper clearly shows. So the problem with this is basically what they have done is they used a standard biofilm model appropriate for modeling biofilm growth in the flow cell and use this as a model of the gut without any changes to it. And it may give the right conclusions, I don't know, because I haven't tried it with more realistic assumptions. It just goes to show that it's better in science to be first with a simple model than to spend more time developing a more thorough model which is more likely to be correct. Because being first is the most important thing in science, not being correct. Another example. This is...
Can I ask a question?
Yes.
When you say that just because this is a summer school, are you saying it's the most important practically if the impact happens, or are you actually, is that a normative statement?
No, I think this is just how science works nowadays. I didn't go into science because of that. I went into science because I really wanted to understand how nature works. So yeah, but I know science isn't quite the way it should be, I think. Okay, so this is an example of Pseudomonas aeruginosa colony sandwiched between a cover slip and agar layer, so it grows into a monolayer producing pyoverdin, which has a nice property of being fluorescent, so you can directly visualize it in the microscope. And you can see here that there are some cells very bright. They are full of pyoverdin. Neighboring cells have sometimes very little pyoverdin. Sometimes the bright cells are at the edge, sometimes they're in the middle, and generally there's more pyroverdin in the center of the colony, but there's a lot of variation between the different cells, as you can clearly see. And another image from the supplementary information, you actually have the brightest cells, a few bright cells at the edge here, and very little bright cells in the middle of the colony. And these are planktonic cells harvested by filtering onto the surface, and you see some very bright cells and some less bright cells. Do you see that, or should we turn the lights on? But maybe you fall asleep if we turn the light off. So you can see there's a lot of variation between individual cells, of course kind of ideal for individual-based modeling. And you can also see in this pedigree that there are these bursts of pyroverdin production for some time, and then it fades out again. So there's some autocorrelation in time in these data, and you have, I think you can see that these are bursts of production. So what they've shown is that if you look at the difference in concentration between the focal cell and the neighbors, and the change in concentration over time, you have this linear relationship. And you can explain this with a model assuming that the pyroverdin is swapped between the neighboring cells. So if the focal cell has more pyroverdin, it gives off pyroverdin to its neighbors, or the other way around. And that can explain these data. And that's kind of fine. So they end up with an equation where they have lumped, I'm not against lumping things, but they have lumped all the processes that decrease concentration into one linear term here. So this lambda includes export, dilution by growth, decay, and some positive feedback that they also consider. So this isn't wrong. It basically shows that you can simplify the system. You can make sense of the data by assuming that the cells just swap the pyroverdin with each other, and there is no pyroverdin in the environment at all. That explains the results. But of course you know that pyroverdin is evolved as a siderophore to go out in the environment and capture iron and bring it back. That's the purpose of this thing. So they are basically assuming that the pyroverdin doesn't diffuse into the environment and is taken up by the cells, but it goes directly between the cells. So this is the way they present the data. It's not actually the way they make the model, because they start with a more complex model, and this is the lumped and simplified version which they then use to identify the parameters. So what they say in the abstract is we quantitatively explain the formation of this gradient by local exchange between contacting cells rather than by global diffusion of pyroverdin. Yes, you can explain this, but this model is then limited to a situation where cells actually have neighbors with which they can swap the pyroverdin, and it pretends that this is what actually happens. If they would present it in the paper in a way that they say, okay, this is a simplification, we've lumped all the processes into those which increase and decrease pyroverdin concentration in the cell, we don't believe that this is actually mechanistically happening, then that's fine. But they kind of don't present it in that way. They could use a more general export, diffusion, import mechanism. They would get the same results, but it wouldn't be limited to this closely packed cells, and would also be appropriate for the planktonic culture studies that they've also done. But obviously this model with neighbors will never predict any other situation where you don't have neighbors, but the pyroverdin production will also happen in a single cell without any neighbors. And of course, as I've said already, the siderophore really is there to go into the environment and extract iron. And they don't explain the bursts of production, which may have to do with the positive feedback, which they have lumped into a linear term, so kind of gotten rid of it. Okay, so it's another example of a paper which looks cool, and it is I mean, it's not bad, but it simplifies too much. And you're simplifying the system to a mechanism which you know isn't correct, because the cells do not actually swap pyroverdin. They have an export and an import, and the pyroverdin is exported by one cell, diffuses around in the environment, and imported by another cell. There is really not a mechanism that the model mechanism is really not existing. So I want to finish with this quote, which is maybe 100 years old, by Kayal in his book Advice to a Young Investigator, which is maybe worth reading. I wished I had read it when I was starting science. What he basically says is we should not be attached to our own ideas, but treat them as prosecutor, not defense attorney. So be critical about your own ideas, your own assumptions, your own work, and that of others. And because it's a summer school, I thought I'll give you some references that I find very useful, which I didn't have too much time to talk about. This paper here, Do Simple Models Lead to Generality in Ecology? It's a very interesting paper that goes in depth into the question of generality, simplicity, and realism of models that came out of a workshop. So the list of authors is something like 15 or so. That's a very interesting paper. Also the other paper with Volker Grimm involved, where they argue that there is an intermediate complexity of the model as an optimal for agent-based modeling. This is a book chapter which you can read if you want to get some information on chemostats and biofilms in a relatively condensed format, and the mathematics of modeling chemostats and biofilms. And this is another interesting paper I recently read. Accurate Descriptions of Our Pathetic Thinking is his take on models. Very, very interesting to read. Thanks very much for your attention.
Any questions or comments?
Yeah.
So you defined individual-based modelling as a set of rules for each individual, and then also how the individual is influenced by its environment, right? So the individual shouldn't have any global information.
Yeah.
What if someone wraps up that global information into some variable of the environment, and then they can pass that back to the individual, and it will still qualify as an individual-based model?
If the individual could potentially sense that information, yes. But the question is, can an individual sense that information outside its cell surface?
Yeah.
Or would it have to read the newspaper to know about it? Yeah, so the cell cannot sense things like gravity, we think, or height, but it could sense the concentration of some chemical, certainly.
Okay.
But only where it is, not somewhere else. Quorum sensing, okay? Sam talked about it. Cells can't count the number of neighboring cells around them. So the whole idea of quorum sensing is that what cells actually can sense is the concentration of the signal. And as Sam had explained, it is not affected only by the number of cells producing the signal, but also by the mass transfer limitation, okay? And also, third thing that Sam didn't mention, but is, I think, quite important, the concentration of the signal is strongly dependent on the clustering of cells. So if you have cells with the same cell density, but they are all clustered, is a very different story to when the cells are more randomly distributed and not clustered. So what the cell can sense is the concentration of the signal, and what inference it can make from that is a big question. It doesn't because there is no simple mapping between signal concentration and cell density, the cell sensing a certain signal concentration cannot tell whether that concentration is there because of many cells producing or lack of diffusion or whatever. Sorry.
So the aging stuff, I guess it must depend must depend a lot on how the aggregates, the misfolded proteins or whatever, are accumulating. So if there's some sort of autocatalytic process, so one misfolded protein will influence and misfold another, then I'd guess that segregation would be more important.
Yeah, so segregation will depend on the fact that you have large enough protein aggregates, which will then tend to end up at the old pole cell. So aggregation is necessary for this damage segregation, and aggregation there are papers showing that aggregation is actually an active process, at least in the budding yeast. So it seems the idea is that by aggregation you're detoxifying to some extent the denatured proteins. By having just one blob of aggregate is better than having lots of denatured proteins floating around in the cell, and this may actually be an active process. But certainly it would the bigger the aggregate is, the quicker it would grow.
So they do expand. They can expand exponentially.
They do expand. Whether I don't know whether there are data to show that they expand exponentially, but yeah, you would expect this, but I don't know whether that's been looked at. Yeah.
So in the last example that you gave, I'm not familiar with any of that, but I would like to ask maybe a naive but perhaps practical question about it. So if I understand, part of the criticism was that they didn't incorporate in the model something that we know to be part of the process. But I'm wondering if, depending on what they were trying to do with that model, or what kind of questions or whatever was the purpose of having that model, maybe that would be already a good enough description of what happens, even if it's inaccurate. So, like I said,
yes, you're right. It would depend on the purpose for which they did this, and to some extent, the model is fit for that purpose. But what they actually do is they start with a model that they know that there is export, diffusion in the environment, and import, and production and decay. They start with these processes. So they know these processes exist, but they lump it together, and then the rest of the paper is written in a way that you think, as a reader, that they actually think the mechanism is exchange between cells. So it's more a presentation issue. If you read the very last bit in the methods section, which nowadays is always at the back in small print as if it's not important, and you shouldn't really read this, so at the very end of the paper you see the original full equation, which they've simplified by lumping all processes together, as I've explained. But the problem really with the paper, I think, is that they then make the reader believe that it's actually an exchange process between the cells. I mean, it's in the abstract, it's in the title of the paper, so the impression you get from reading the paper is different from the sort of the starting point of the model. So that is my problem with that paper.
One more question.
Yes.
So you did mention something about how you would like parameterize an individual-based model. But I was wondering if it was difficult to do that, because microbial growth parameters are measured in a way that already incorporates the maybe some emergent properties. Like if you make, you've measured those sort of Km and the Vmax parameters on a community that already would incorporate some emergent.
Yeah, that would be problematic. So this is a problem if you're measuring the kinetics of a cluster of cells that incorporates already the mass transfer into the cluster and within the cluster. So the kinetics you measure are not representative of the kinetics of the individual cell. And for the individual-based model, you need to specify the kinetics of the individual, because a model will also include anyway the diffusion of the compound to the cell. By taking measurements which are combined mass transfer limitation with cell kinetics, this would be wrong. Okay, sometimes if this is the only data you have, that's better than no data, but you shouldn't be under any illusions that this is a good way to parameterize a model. And that means you cannot be certain about the parameters, so you should certainly look at the sensitivity of those parameters for your results. Okay.
Yeah, so thanks, Jan. So just before we thank Jan again, I've got two announcements. First announcement is about the conference dinner. So it appears that some of the helpful things on your badge are actually not entirely correct. So the admin staff have made a definitive list of who they think is going to conference dinner, and this list is on the table out there. So if you could sort of check the list and see if it agrees with what you thought, and if it doesn't, maybe go and ask at reception. That would be helpful. And the different coloured shading on the list doesn't mean anything, so you should just ignore that. And that was the first announcement. And the second announcement is now we have a wine reception, hopefully, and also poster session. So let's enjoy that. And now let's thank Jan again.